Hey guys, I will start with immunoglobulins, structure and function. Immunoglobulin, also known as antibodies, or these antibodies are secreted by B cells. Antibodies are the antigen binding glycoproteins synthesized by B cells. ये जो एंटीबॉडीज हैं फर्दर एग्जिस्ट इन टू फॉर्म्स soluble form or membrane bounded antibodies forms. Soluble antibodies are secreted by B cells and membrane bounded antibodies are present on the B cell membrane. These both types of antibodies, soluble or membrane bounded, are differ from each other at carboxylic terminal domain. And it means they are differ from each other at carboxylic terminal domain. Soluble antibodies are hydrophilic amino acid sequence at carboxylic terminal. Further, we explain basic structure of antibodies molecule. Antibody molecule is Y-shaped structure. It has two identical antigen binding in sites on tip of each arm of Y-shaped. Look that antibody is a Y-shaped structure. It consists two sites antigen binding site at the tip of the antibody molecule. So antibody is a bivalent. ठीक. It consists of four polypeptides. It consists four polypeptides, two light chains or two heavy chains. Light chain further consist 220 amino acids, where heavy chains consist 440 amino acids. Both covalently linked by disulfide bonds, which means that light chain and heavy chain both uh, joints with disulfide bond covalently. Further, we explain both light and heavy chain consist amino terminal variable regions and carboxylic terminal constant regions. Further, it is an amino terminal or it is an carboxylic terminal. Further, variable. I can explain in letter variable reasons. Variable reasons, one heavy chain and adjoining variable region of one light chain from an antigen binding site. Light chain or heavy chain. Both variable regions are joined together to form an antigen binding site. For the light chain, we explain light chain. Light chain is two subclasses. Light chain consists two subclasses, kappa and lambda. It consists always either both kappa and both lambda, never one of each. When an antibody consists a light chain, it can be both have kappa kappa or both have lambda lambda light chain. In we explain that an antibody consists that two heavy chains and two light chains. So both the light chains can be a kappa kappa or can be lambda lambda. Further, we explain heavy chain. Heavy chain consists five different subclasses: alpha, delta, gamma, E or mu. These subclasses explain which type of antibodies are there. Whether alpha to IgA, then delta IgG, then gamma IG, uh, sorry, delta IgD, gamma IgG, and so on. These are called isotopes. These are called isotopes. Further, both heavy chain is antibody are identical. Both ये जो हैवी चेन्स प्रेजेंट होती हैं एंटीबॉडीज में, दो हैवी चेन्स प्रेजेंट होती हैं विच एक्सप्लेन दैट विच टाइप ऑफ एंटीबॉडीज आर प्रेजेंट इन आवर बॉडी. For example, IgG class, it consists 2 kappa or 2 lambda, 2 gamma chain. Gamma chains explain that it is an IgG type of antibody. Heavy chain of a given antibody molecule determine the class of that antibody. I explain that कि जो heavy chains हैं वो explain करते हैं which type of class of antibody is present. Further, alpha and gamma heavy chain further classify in two subclasses. Alpha means IgA, alpha 1, alpha 2 means that IgA1 or IgA2. Then gamma IgG. means that gamma 1, 2, 3 or 4. It can be classified in subclassify into 4 classes. Then antibody molecule. I will explain some properties about antibodies. Antibody molecule are flexible. Flexible कैसे है वो explain है. Flexibility compressed largely due to presence of Hing region. Hing region is present in between at that location. Hing region is present. Located between CH1 and CH2 in certain isotopes, carbon 1 and carbon 2 in certain isotopes. It consists from 10 to more than 60 amino acid residue. Hing region consists 10 to 60 amino acids residues. The alpha, gamma and delta heavy chain contain the Hing region, which is rich in proline and cysteine residues, giving IgG, IgD, IgA flexibility. It means that the Hing region in Ig, G, D or A consists proline and cysteine residues, which gives the flexibility to the antibody molecule. Between the two Fab are more Y-shaped antibody molecules. Means I will explain. The antibody molecules consist two regions. One is Fab and one is crystallized FC region. Further, antibodies I explain in soluble or membrane boundaries. Antibodies. Soluble antibodies have hydrophilic amino acid sequence at carboxylic terminal domain. Soluble antibodies के examples हैं IgA, G और E. Membrane bounded antibodies के example हैं IgM, IgD. Carboxylic terminal domain contain 3 regions. यहाँ पे carboxylic terminal region क्या consist करता है? हाइड्रोफिलिक अमीनो एसिड्स, जबकि यहाँ पे कार्बोक्सिलिक टर्मिनल कंसिस्ट कर रहा है 3 रीजन. एंड एक्स्ट्रा सेलुलर हाइड्रोफिलिक स्पेसर सीक्वेंस, हाइड्रोफोबिक ट्रांसमेम्ब्रेन सीक्वेंस एंड ए शॉर्ट साइटोप्लाज्मिक टेल. फर्दर डिड्यूशन ऑफ एंटीबॉडी स्ट्रक्चर. अब किन किन ने एंटीबॉडी स्ट्रक्चर के बारे में क्या क्या बताया, डिफरेंट पॉइंट ऑफ व्यू थे. पोर्टर और एडलमैन indicate the basic structure of antibody molecule, but their approaches were different. इन्होंने antibody molecules के बारे में बताया, but दोनों की different approaches थी. इन्होंने क्या किया, पोर्टर थे, उन्होंने पेपिन डाइजेशन करवाया, जिससे जो एंटीबॉडी मॉलिक्यूल्स हैं वो 3 फ्रेगमेंट्स में डिवाइड हो जाती है. जबकि एक तीसरे थे निसॉन्फ, उन्होंने जब पेप्सिन डाइजेशन करवाया तो जो एंटीबॉडी मॉलिक्यूल है वो 2 के अंदर डाइजेस्ट हो जाता है. जो FC फ्रेगमेंट है, मतलब क्रिस्टलाइजेशन फ्रेगमेंट जो है वो मल्टीपल फ्रेगमेंट्स के अंदर टूट जाती है, ब्रेक डाउन हो जाती है, सो दैट दिस फ्रेगमेंट आर नॉट रिकवर्ड. फर्दर और इडमैन जो थी रिड्यूस द एंटीबॉडी मॉलिक्यूल IgG यूजिंग बीटा मेरा कैप्टो ethanol in order to break disulfide bonds. Hey guys, for today it's over, and I will teach next topic at the next time, but I will explain all the topic of today in repeat. Immunoglobulin are the antibodies. They are secreted by B cells. They consist antigen binding sites. Further, एंटीबॉडी जो मॉलिक्यूल्स था वो मैंने बताया कि 5 शिफ्ट स्ट्रक्चर है. इट कंसिस्ट अमीनो टर्मिनल और कार्बोक्सिलिक टर्मिनल. और फर्दर एंटीबॉडी मॉलिक्यूल सोलेबल होता है, मेम्ब्रेन बाउंडेड होता है. सोलेबल जो होता है वो सिक्रेट किया जाता है बी सेल्स के द्वारा, और मेम्ब्रेन बाउंडेड होता है वो सब प्रेजेंट होते हैं ऑन बी सेल मेम्ब्रेन्स पे. फर्दर जो एंटीबॉडी मॉलिक्यूल है वो कंसिस्ट करता है हिंग रीजन विच कंसिस्ट 10 to 60 अमीनो एसिड रेसिड्यूज. दे प्रोवाइड फ्लेक्सिबिलिटी. फर्दर सोलेबल एंटीबॉडीज और मेम्ब्रेन बाउंडेड एंटीबॉडीज के एग्जाम्पल. सोलेबल एंटीबॉडीज जो थे उनके एग्जाम्पल्स आते हैं IgA, GE. और मेम्ब्रेन बाउंडेड एंटीबॉडीज थे उनके एग्जाम्पल थे IgM और IgD. Further, light chain, heavy chain, explain. Light chain consists further divided into two subclasses, L kappa and gamma. Each antibody molecules either both kappa or both lambda, neither one of each. Heavy chain further subdivided into five subclasses, alpha, delta, gamma, E, mu. Heavy chains determine the which class of the antibodies function. So that's all for today. Students, I expected that you will support me and subscribe my channel. Thank you so much.